It's a really my great pleasure to attend this event sponsored by RWD. And we all know that it's been a very difficult year for science exchange, especially between the United States and China. So this is a really wonderful opportunity for me to sort of talk about my work in the last couple of years. And then, first of all, I'm not really an expert to define anything. Let's say what is the vascular path to dementia, but I want to open this discussion with all of you guys to see, do we really have a unique sort of perspective on that, and how can we work together to achieve that goal? So first, my lab is in USC, the University of Southern California, and we are located in Los Angeles. And this is the picture of downtown. Okay, you guys can, okay, this is downtown LA. So our campus is probably around here. And my research is mainly focused on the Alzheimer's. And then we are located in this Zuka Neurogenetic Institute here. And then we are affiliated with the Alzheimer Research Center as well, and then the neuroscience graduate program. And at USC, we have multiple centers and institutes really working together on solving this puzzle regarding the Alzheimer's disease, including the neuro sort of imaging centers as well as the therapeutic research center at San Diego. And so back when I was in graduate school, sort of I started as in neuroscience, and at that time and until now, I think most of us probably really interested is to solve why and how the brain is working. And then our thinking and everything, all the cognitive function is conducted through this dynamic sort of neural networks, right? But then the problem is that the neurons have to use tons of energy, and this energy is supported by this, the vascular systems. It's estimated that 20% of the energy goes to the brain. And besides of this energy supply, that we know that the neural cells, including neurons and glia cells, are formed sort of really closely to the vasculature, and this sort of position as well as sort of functional sort of orchestra is now considered as called the Neurovascular Unit. This is a term to describe the close sort of relationship between the neural cells and the vascular systems. And besides all these two, and now we also know that there is an active sort of CSF flow from the sort of choroidal plexus towards and going to the drainage through the brain's lymphatic systems. And if we consider the vascular systems, I would think that it definitely need to include the blood vessels as well as the lymphatic systems, and so that, in my opinion, I think most of you guys will agree, the healthy brain really needs is a healthy sort of vascular systems, right? So in my lab, basically we study the crosstalk between the vascular cells and the neurons and glia cells. We culture them sort of independently and mix them in the kind of mixed culture and determine their sort of crosstalks. And then we also work heavily on the animal models to look for their sort of different levels of pathology. This is a sort of top pathology. This is a vascular pathology, showing that this is a stretched vessel has a leakage. And we also, look at a genetic profiling as well as developing, gene therapy tools such as AAV to sort of amend, this disease, right? And so during the last couple of years, RWD has really been really helpful for us. They help us to establish the TBI, traumatic brain injuries, setup, as well as sort of the laser speckle systems to measure the blood flows. And today I probably won't be able to go much detail into this, but they've been offering us is really sort of custom-made platforms as well as sort of high quality instruments, and it really help us to establish our program. Now, let's go back to the question, that we having a problem with dementia. And this is actually is coming from a good side of the society, which is that we're all living much longer now. That, but the problem is once we all pass 65 years old, then the risk of dementia has really increased. And this is happening first in the European, and then to American, and to Asia, and now probably going to Africa as well, right? And so there's tons of sort of age-related dementia and neurodegenerative disease. There are Alzheimer's disease, vascular dementia, frontotemporal dementia, Parkinson, LS, Huntington's, and I think there's at least about 50 of them. But today, because of time, probably only able to touch base on the AD and the vascular dementia a little bit. And then, but the take-home message is that most of those disease has vascular complications, right? And so let's go to the first patient. So I think it's in the beginning of the 20th century that Dr. Alzheimer reported the first patient, August, right? And then it was basically, you can still find this patient's documents, document file. It's very simple, that describes the observation of the first patient as well. Later on, he reported postmortem studies showing there's the plaque and tau, right? And that is the first case. But now we have almost 500,000 every year, just in the United States. And the patient is going through very complex diagnosis, including, let's say, behavior assessment and cognitive assessment, as well as, neuroimaging and biomarkers. And potentially, it has to be validated in the end through the postmortem studies, right? And although we have now different technologies and methodologies to diagnose the patient, but in terms of treatment, we're not offering too much comparing like 120 years ago. The patient is still suffer, and this sort of progressive disease course, and this leads to the sort of Alzheimer's sort of challenge. And now in the United States, it's about probably 5 million people with Alzheimer's. In the worldwide, it's probably 50 million, and then the cost of that healthcare in the United States is more than $1 trillion. And despite of this kind of challenge, the pharmaceutical companies still having really struggle to find the cure, right? And then, so the researchers, including us, is going back and trying to think about this whole science and biology behind it. And we know that after the first discovery, it takes about like 80 years for the scientists to discover that the pathology happening in the patient, which is, let's say, the amyloid plaque, is coming from the very small peptide called amyloid beta. And it is cleaved from this amyloid precursor proteins by 3 different proteins: the alpha secretase, beta secretase, and gamma secretase. And eventually generate this 40 or 42 amino acid peptide. And this peptide is prone to aggregation, eventually forms the plaques in the brain, right? And there's a different pathology called the neurofibril tangle that is actually formed by another protein called tau protein. And the tau is, once it's hyperphosphated, it goes to, protease cleavage as well as conformational change, and eventually forms the aggregation inside the neurons. And these two are basically, the puzzles are really well solved. And that leads to the amyloid hypothesis, as well as sort of targeted treatment, right? And ideally, it's very simple. That if we can prevent the cleavage, let's say beta secretase and gamma secretase, and then we can block the production of these amyloid beta. And if we can design antibodies or sort of small molecules that prevent the aggregation, then we should also able to prevent the plaque formation in the brain. And this has been, all those treatments have been proven working in the preclinical models. Somehow it doesn't work in the patient, right? At least in the phase 3 clinical trials. And the same happening for the tau treatments. And there's a lot of inhibitors, inhibitor caspase, and then the kinase, phosphorylation, as well as acetylation, and then antibodies as well for the tau. But so far, none of these sort of trials are been really working. There's still probably 200 trials targeting the amyloid, about 50 targeting the tau is ongoing, but we don't know how promising they'll eventually be. So that leads to sort of the whole sort of field and the society to rethink about this problem. And a simple explanation is the Alzheimer's must have a very complex etiology, which is probably same to, let's say, other metabolic disease like diabetes, as well as hypertension and others, right? So besides the memory function, neuronal dysfunction, as well as the amyloid and tau, there is a complex genetic backgrounds, and then other complications as vascular, and then neuroinflammation. And most of us know that the lifestyles, environmental factors contributes to the disease as well, right? So as a scientist, we always go back to, let's say, what's the genetics, right? And then when I was in graduate school, we always hear, genetics never lies, right? And that actually is the case, that we look at the sort of familial cases, which means that the families has mutations, right? If you trace back in the sequence, and it turned out that there's 3 genes that are commonly sort of mutated in those familial cases. These are the APP, which is the precursor protein of the amyloid beta, the presenting 1 and 2. These are parts of the components of the gamma-3 secretase. So mutations in those 3 genes eventually leads to sort of upregulation of the amyloid production, right? However, over 90% of cases are not familial, they are sporadic. And these are the patients who do not carry those mutations. And that's the reason that, if we design that the amyloid and tau therapies for these familial cases, the chance of them working in the sporadic cases are much lower, right? And for these patients, they do not have sort of dominant mutations, but they are more influenced by risk genes as well as environmental factors, right? And you guys probably familiar with this, Manhattan plot. This is coming from about 2 decades of association study, to identify the genes that link to the Alzheimer's disease. And this is a method that came out, last year, and that is from almost 100,000 patients. And you can see that they identify about 25 different genes that highly associated with the late onset Alzheimer's disease, which is sporadic cases. And among them, you probably know that ApoE, right? ApoE is most of the significant risk factor for Alzheimer's disease. And one allele of ApoE4 increase the risk by, let me see, there's some chats. Oh, sorry, anyway. So that one copy of ApoE4, increased about 4-fold of the likelihood of developing AD, and also shorten the sort of disease onset about 7 years. Right? And then you can also think that this is a vast list of genes. Some of the genes plays a role in the immune system, such as CR1, right? And then it's a SPL1, and those are sort of considered as the immune genes. And some of them actually plays roles in the vascular systems. ApoE is one of the atherosclerosis gene, right? And then ADAM10, ADAM10. Those are sort of the alpha secretase that plays a role in the vascular systems. And today we're really going to focus using PICOM as an example to illustrate how those risk genes linked to Alzheimer's and potentially is through the vascular path. So PICOM is a clathrin assembly protein. That means that it plays a role in the endocytosis process, right? So the first step we did was basically to stain the PICOM in the postmortem tissue from humans. This is from the cortex area. And you can see that in the control samples, the PICOM is really mainly in the sort of vessels, right? When you look at in the AD, in the later stage AD, you see the vessels has very little PICOM stainings. But the rest cells, if you look at these guys, those are sort of neuronal cells. They seem to be still have pretty good level of PICOM, suggests there is a selective sort of reduction of the PICOM in the AD. And that's also can be validated by Western blot. If we isolate the microvessels from AD patient and we see the pickup level goes down, right? And that can be also validated in a different region, which is hippocampus, that responsible for the learning and memory, right? That if we compare in the control, you can see that the pickup, which is green, has a lot of sort of staining in the vessels, right? But when you look at the sort of the AD cases, the green signal in the vessels is really reduced, suggests that there's selective sort of reduction of the pickup in vessels. Now, if we quantify that the pickup vessel areas, right, we can see there is a strong correlation that when you have a lower sort of pickup in the vessel, you have higher amyloid load, as well you have sort of more advanced Alzheimer's stage based on the BRCA sort of criteria. And also that worst sort of cognitive functions based on the MLSC SE so score, as well as the CDR reading. So suggests that sort of there is a selective reduction in pickup that is associated with AD progression, right? So why is that? So at that time, we didn't have the sort of conditional knockout. So we utilized the heterozygous sort of 50% reduction of pickup model, which is pickup plus minus, and cross with the Alzheimer model carrying the Swedish mutation of NTP. This is also known as TG2576. So we realized that after we reduce pickup by 50%, we see a greater sort of upregulation of the amyloid pathology, both in the hippocampus and the cortex. And the mouse also behave much worse in terms of different kind of learning memory tests, including nesting, burrowing, and novel object location and recognition tests, suggest that just by reducing the pickup, the mouse are more vulnerable to the amyloid pathogenesis. Now, how do we know that the vascular pickup plays a role? So we designed a rescue sort of experiment to put the pickup back to the vessels by using the AAV. And this is a flux AAV, so that it only works with a vascular cream, which is type 2, using a type 2 promoter. So we can see that using this approach, we can basically re-express pickup in the vasculature only, right? And then when we look at it, is that we inject it in the hippocampus area. And you can see that just by injecting the virus to deliver the pickup back to the vessels, it reduces the amyloid pathology in the injected hippocampus area, but not in the uninjected cortex area. Suggests that there is a very sort of selective rescue in just using the vascular pickups. Right? Now, the mechanism of that is that pickup really binds to a receptor. This is called low density lipoprotein receptor, like protein 1. This is a major receptor for amyloid beta in the brain, as well as ApoE, right? And then we're able to map the binding. That it turns out to be this NPXY domain that is responsible for the binding of the LRP1 to the pickup. If we mutated those residues here, you can see there's no longer a binding, right? And then we can also mimic the sort of the blood-brain barrier in the dish. Right? In this case, we play the endothelial cells in the transwell, and they form sort of very good sort of blood-brain barrier kind of properties. And we can identify that the association between the pickup, LRP1, and amyloid beta, and in those preparations. And by doing so, we're able to sort of monitor the molecular events in terms of the association and the transport kind of events. And one of the techniques we use is called the proximity ligation that determines the close interaction between two molecules, right? In this case, we can detect the transient sort of interaction between clathrin and LRP1, but much prolonged interaction between pickup and LRP1. And that suggests that a pickup is not like a clathrin. It does not really dissociate from vessels, endosomes. It actually travels along with the cargo, which is the LRP1 and amyloid beta, and drives the cargo through two different processes, which is the early endosome, which is Rev5 positive early endosome, and Rev11 positive sort of sorting endosome. But it does not go to the Rib7 positive some linear adenosome, all goes to the lysosome. So this gives us a sort of picture. We really sort of put in this kind of diagram that once the amyloid beta and LRP1 forms the interaction, it goes through the endocytosis pathway, which is driven by the pickup, goes to the sorting, and then eventually goes out towards the blood side. And this side is the brain where the amyloid beta is produced, right? And this is just a small piece in this kind of clearance map, right? It's been sort of studied about like 2 or 3 decades now that in the brain that your blood-brain barrier has the clearance mechanism that receptor is able to recognize the ligands in the brain, particularly amyloid beta, and pump them out towards the blood, and then eventually goes to the peripheral for degradation, right? In the Alzheimer case, there's LRP1, LRP2, and then VLDLR. And that works with different sort of the risk genes, such as pickup, right, and clustering, and also works with ApoE. And this kind of mechanism eventually leads to a very effective sort of clearance of the amyloid beta. But somehow that when you have mutations or genetic sort of variants, such as E2 and E3 variants compared to E4, then you basically shifted the clearance of the sort of efficacy. It leads to reduce the clearance, right? So, but then this clearance mechanism is just a sort of tip of iceberg. And if we look at the blood-brain barrier, there is a very complex sort of transport systems that is built in through the evolution as well as the development, right? So we know that there's ion channels. There's transporters for carbohydrates, the amino acids, and fatty acid, and others, and proteins as well, right? And there's also drug transporters to keep the drugs away from the brain. And then for the Alzheimer cases, we know that LRP1, that we just introduced, that it pumps the amyloid beta to the blood. And there's another receptor called RAGE, actually is doing the opposite. So during the vascular inflammation, RAGE goes up. That also leads to pumping back the amyloid beta back to the brain, so that is better for the brain health, right? So a few things I want to point out is that GLUT1 that pumps the glucose into the brain. So you see that in the amyloid patient that they usually have reduced the glucose uptake. That's likely because of a reduction in the glucose 1 transporters on the blood-brain barrier. And another one I want to point out is this called the MFSFD2A. So this is a transporter for the omega-3 fatty acids. So when you have a reduction in this, you basically have less omega-3 fatty acid in the brain. That leads to a microcephaly as well as reduced cognition as well, right? And on the other side is that we now know that this is called parasites, that a cell next to the blood vessels, and it also has tons of transporters, which is really unknown for now. How do they actually play any roles in the brain health as well as Alzheimer's disease, right? Now I want sort of a little bit switch gears. So we talk about how the vascular system has the clearance pathway that actually help to maintain the amyloid sort of homeostasis in the brain. And then most of you guys know that there is a term called vascular dementia. There's always been talking about how does that link to Alzheimer's disease, or how does that is different from Alzheimer's disease. But let's take it back, is that there is early some awareness that it probably occurs that in the beginning of the 20th century, before the discovery of the Alzheimer's, that the dementia are really coming from the vascular aspect, because at that point that the life expectancy is probably around 50s, that if the dementia occurring, usually is caused by a stroke or something, right? And after about 100 years to the late 90s, now everybody believes that in the elderly, which would be the older populations, the dementia really is because of Alzheimer's disease, right? And at that time, they think that there's nothing to do with the vascular dementia, but that view has been shifting after in particularly in this century. And that reason is that we know that really based on the clinical studies that in the stroke, you will have a huge infarct, right? That are going to be specifically affecting in one of the sort of brain areas that links, let's say, to language, to motor functions, or cognitions. This can be easily identified through neuroimaging and compare the imaging with the cognitive functions, right? That is vascular dementia. The problem is that there is a huge population of Alzheimer's disease patients. They do not carry this big stroke, right? Or infarct. They do carry those kind of smaller lesions. But these are also associated vascular incidents, such as, this is called silent infarct, right? And there's always, well as now we know, this is very common. It's called diffuse white matter sort of disease or damage that is commonly detected in age populations, particularly Alzheimer's patients. Now, these are the populations that have been overlooked because of these leads to, does not lead to immediate sort of functional deficits, right? It's being overlooked. Another aspect is that, you can take postmortem tissues and look for their, pathologies. And there's multiple studies has, really show that maybe only 20% of the patient populations are pure, that they only have amyloid or tau pathology that does not have vascular pathology. But the majority, which is almost 80%, the AD postmortem samples, they have both amyloid and tau, as well as strong vascular pathology. Now, this population is considered called mixed Alzheimer's disease. It mixed with vascular pathology as well as amyloid pathology, right? Now, this is now a new term called vascular cognitive impairment and dementia. So it demonstrated how big an overlap is between the vascular dementia and the Alzheimer's disease. Now, this is something that we really puzzles us. Why is that? So the answer to address is to understand is that the vascular contribution in the Alzheimer's disease. That leads to, we have to go back and think about what is the vascular system, how does the system work, and then how does that contribute, right? So I mentioned to you guys earlier is about neurovascular unit here, right? If you look at it more carefully, is that this is the top of the skull. The arterial goes down, and in the arterioles that you will see that this is endothelium, right? There's tons of the different types of components, including the smooth muscle cells and endothelial cells, astrocytes and basement membranes and other things, right? And then it becomes branch out, becomes the capillaries, and then eventually capillary merge together, become the venule and the veins. And then the cell composition is totally different at different segments. And at least by single cell studies, that we can identify that there's about 3 different cell types in endothelial cells and 2 to 3 different parasite types, and probably 2 different SMC types. These different cell types are really work together, with the neurons and glial cells differently. So let's say the endothelial cells, they will work with the neurons through the VEGF and other signalings. The parasites, they have also growth factors that specifically expressed in the parasites and work with the neurons, right? And then this crosstalk is sort of relatively a new area for us to understand how that actually work. And so today, due to time, I can only probably take one example, which I will pick is the parasites. The parasites is the cells grow next to the blood vessels and at level of the capillary level. So if you take it EM, the electron microscopy scanning, you will see. So this is the cell body, and these are the process. They basically wrap around the vessels, and you can also identify it by immunostaining uses the different markers such as the PDGF-beta receptor. And in the mouse that are lacking parasites, which is the, let's say, red model or PDGF-beta plus minus model, you will see there is a problem with blood-brain barrier. So that is a classic experiment that when you inject the blue dyes in the bloodstreams, this dye will not stay in the brain after perfusion. But when the blood-brain barrier is sort of impaired, or there's a breakdown to the blood-brain barrier, then you see the dye stays in the brain. You can use a different dye, let's say fluorescence dye, let's say this is red dye, so you see red signals in the brain as well, and this is another dye. Suggests that in those mice models, there's a problem with the blood-brain barrier. Now, so recently we're actually able to generate a Cree line using a complex strategy using the two promoters, PDGF beta and CSPG. Now I'll skip on why the design, and then we'll just say that we do have a specific promoter now to drive the Cree in the parasites, and you can see the red ones are the reporter, the T2 tomato reporter, driven by this this parasite Cree line. There is specifically expression of this reporter in the parasites, but not on the smooth muscle cells. You can see this is the arterial comes in. There's no T tomato signal at all. The signals are really on the capillary levels. And we can probably achieve easily with about 70% of sort of labeling with this model. And using this model, we're basically able to cross to a Cree-dependent DTR receptor mice. And this receptor will basically is a diphtheria toxin receptor, right? So once the receptor is expressed in the parasites, we can give the mice, diphtheria toxin. The diphtheria toxin will basically kill all the parasites in this mice. And this is it definitely is the case. This is just with one injection. After 2 days, you can see the apoptosis markers coming up in the parasites. And you can also see the reduction of the parasite markers in the brain. And we did more quantification that within 6 or 7 days, all these T tomato positive parasites, are gone. Suggests that we can really selectively ablate those parasites using these models. Now, we can follow these mice with neuroimaging different techniques. And at least, MRI neuroimaging tell us that there is about 50% reduction in the blood flow in both hippocampus and the cortex after parasite ablation. Suggests the parasites is important to maintain the blood flow in the brain. At the same time, we can measure the blood-brain barrier sort of permeability using the this is DTI neuroimaging. So you're using a cadaveric tracers, and the tracer will enter the brain from the bloodstreams, and that can be monitored as the K-trans. The K-trans shows that there's a sort of increase in permeability after we ablate parasites that occurs, after 3 to 6 days towards it gets worse over the 2-week period. And this K-trans can be, sort of validated through the histology. And in this case, we're using IgG. It's a theory. It is the antibody that it's in the blood. And we can see that now the IgG basically leak out into the brain and stays in the brain, as indication of the blood-brain barrier being sort of breached. And so we have demonstrated to you guys that the vascular phenotype occurs between 3 to sort of 12 days. Now that when we look at 12 days, we do not see any sort of cognitive function change. However, when we look another 2 weeks later, we do see reduction in the combinations. This is a memory test, right? And this is a different test showing that they have problem recognizing sort of new objects. Now this change is really associated with sort of neural change. And at 2 weeks post that vascular sort of phenotypes, we see a reduction in this sort of the number of neurons in the hippocampus and the cortex, as well as reduction in the axonal densities that suggests that the neuronal changes are secondary to the vascular changes that drives the cognitive functions. So now, excuse me. So we know parasite plays multiple roles in regulating blood-brain barriers, as well as angiogenesis. Through our studies, we know that it also regulates the blood flows. And these are very key functions, but then it also has neuroinflammation response that are being reported by other labs, as well as stem cell activities reported by other labs. But when we consider all these functions together, we know that all these functions are going to contribute to neuronal functions, as well as other aspects. For example, that earlier we reported that in the different parasite diffusion model, which is F7, there is a white matter sort of atrophy as well as axonal damages. And then if we cross this PDGF plus minus model with the APP Swedish model, then we can see there's an increase in amyloid pathology as well as appearance of the tau pathology. Now this suggests that parasites really contributes to multiple aspects of the neurodegenerative cascades, including the neural loss as well as amyloid and tau pathology. So this is something where we want to just go back and think about the etiology. So we know that when the vascular incidence happens, it not only contributes to the energy problems, because this can be, as I mentioned, that if you look for the glucose uptake, it usually is greater, at least about 50% reduced in the Alzheimer patient. And these are associated with vascular incidence, including the small infarcts, and then those are microbleeds in the brains. But these are energy perspectives. And we know that when the blood-brain barrier breaks down, there's different aspects. There's leads to the neurodegenerations, right? But now the question is, does that blood-brain barrier breakdown really happens in Alzheimer's disease or not, right? This is a case report even earlier, sort of 2016, showing that if you compare an 80-year-old sort of healthy patient with 60 to 80 years, which is about at least 12 years younger patient, right, with AD, you see that BBB leakage is much higher in the Alzheimer patient compared to the healthy control, right? This is just one case report. So this is a study that conducted at USC in 2015. What they did is basically compare young patient and older healthy controls with mild cognitive impairment, which is considered as early AD, right? So if you compare these, you will see clear sort of difference that in aged populations, there is a worsening of the blood-brain barrier. In the AD, you will see an even worse of the blood-brain barrier than in the hippocampus. And this that indicates that the blood-brain barrier function is diminished in the Alzheimer's case, right? Now, if we look back and we examine all the literatures, we find that if we compare all the familiar mutations, APP, PS1, PS2, you identify probably hundreds of mutations, but you will see a vast majority of these mutations actually also links to the BBB function, problems. And this is through a literature survey, right? Not only, those mutations that you can see, ApoE4 is also linked to the blood-brain barrier dysfunctions. And as I mentioned, there's other neurodegenerative diseases such as LS. Those are mutations associated with barrier dysfunctions. And there's Parkinson's mutations, Huntington mutations, all associated with vascular dysfunctions. And not only these familial cases, there's also sporadic cases that are known to sort of causing sporadic and risk genes also linked to sort of vascular dysfunctions as well. So right now that is based on the human patient data. All these mutations can be recapitulated in the mouse models. For example, that in this case we have the ApoE4 crossed with FAD mutations. And in this mouse model, you can see a really breakdown of blood-brain barrier to this tracer, right? This tracer can be selectively uptaken by the neurons. You can see that's why those showing up by those dots. Those dots indicates the neurons uptaking those tracers in this model, but not in the controls. Suggests that the barrier is globally sort of impaired in this Alzheimer model mice. Okay, so if we think about it, is that now those human data suggest the mutation is linked to it, but how do we know that in the sporadic cases actually they also have this problems, right? We know that there's MI studies now we can apply. There is, DCE, MI, and the T2 star for different sort of proliferating as well as microbleeds, right? And then there's biomarker studies that we can really target on those different sort of molecular signatures. For example, there are RAGE and MMP and cytophenin and LRP1s, right? And then those combined sort of neuroimaging and biomarker study will enable us to look at for a different stage, right? So and these are sort of a few of recent reports showing that if we combine all the data, it suggests that if, in at least a certain population of Alzheimer's case, that the vascular sort of biomarkers, neuroimaging sort of data actually shows earlier changes compared to amyloid and beta and then tau, sort of that. And then amyloid beta and tau biomarkers also sort of precedes the cognitive changes. So that suggests that there's a certain population of Alzheimer's, sporadic late onset Alzheimer's, they are impacted by the vascular change early on in their disease. That is probably that being overlooked in the past, and this is deserve us to really look at it and sort of design treatment for them sort of separately compared to the sort of amyloid and tau sort of driven cases. Now, why is that? So we know that vascular sort of dementia leads to sort of changes in the brain, but there's other sort of risk factors that really drives the Alzheimer's disease. For example, traumatic brain injury, that is number one environmental factor, right? Because the brain injury leads to different changes in the brain, including neural damage, neuroinflammation, as well probably vascular dementia, right? Then, so in the TBI case, that 80% of them are considered mild traumatic brain injury, MTBI. These have been sort of really overlooked in the past. So we be able to establish the model using the RW equipment. In this case, we're going to have a control cortical impact on the intact scalp. That'll produce a small sort of impact in this area, and you can see just one impact results in accumulation of the Evans blue in this region. And we can quantify the Evans blue in this region. You'll see that this is the impact area. There's a breakdown of the barrier to the Evans blue. That's really a result of the impact. The impact also reduced the blood flow by probably 40 to 50%. So this is something, one of the example I want to bring to you guys is that those environmental factors such as brain injuries, as well as, let's say, diabetic cases, they all link to the reduction in the blood flow as well as blood-brain barrier changes. So this is a secondary hit that combined with amyloid eventually probably going to drive the Alzheimer's disease. So quick summary here is that in the second part of talk we discussed is that there's probably independent pathway that occurs in Alzheimer's, where we have the blood-brain barrier breakdown or loss of parasites, right? That leads to different pathways, and that's been shown by different groups. There are groups showing that RBC, that the red blood cells coming out from the blood will generate the ROS in the brain, causing the oxidative sort of stress, right? And earlier we showed that the fibrinogen activates microglia, and then there's albumin that leads to edema, and there's also an antibody come in that causes neuroinflammations. Those pathways are really, independent of amyloid and tau, and can drive this sort of neural damage, that occur not only in stroke and TBI, also in AD and in LS and other things. Now, for the Alzheimer's, there's a specific pathway that we discussed in the first path, which is the sort of the amyloid sort of clearance pathway that in the blood-brain barrier that you have receptors specifically for those amyloid and pumping up them through different pathways to the blood-brain stream for degradation. So that it can be impaired in the Alzheimer's disease as well. Okay, so that's, I think, is addresses probably one of the questions raised earlier is that what is the vascular contributions, particularly in the Alzheimer's disease case, right? Now, as I mentioned earlier, that Alzheimer's disease is only one neurodegeneration, right, cases. There's a lot of others, and including also brain infections such as Zika and COVID, right? So, in my opinion, I think probably hopefully you guys agree is that for all the things to breach into the brain, they have to pass two things. One is the blood-brain barrier, the other is our immunity, right? So all these disease comorbidities such as TBI, diabetes, and then environmental factor lifestyles, really weakens our barriers. And then so the pathogen is going to be easy to pass through. Or that there's also intrinsic signals in the brain that are going to be making the brain more vulnerable to them. So that's why there is a complex etiology in all those kind of neurodegeneration cases. In the Alzheimer's, I think we know much clearer now that what is the amyloid contribution here. The amyloid basically will make the blood-brain barrier much weaker, as well as the evidence suggests that there are risk genes that it also weakens the barrier as well as immunity. So that's something that I think we're going to discuss. But one thing I want to mention is that it's coming from the sort of pre-submitted the questions about tau. So tau is definitely now the very sort of unique aspect of the degenerations. And for the tau, that it also occurs in different aspects. So in the Alzheimer's disease, particularly in FTD, and then the TBI as well, right? In those cases, I think there's a difference. So tau is mainly sort of occur in the neurons. It can potentially be released from neurons and sort of transmitted to different regions. And there's reports showing that the tau can sort of transmit it through the brain circuit as well as through the blood-brain barrier, because there's evidence suggests that once there's local breakdown of the blood-brain barrier, the tau species can now go into the bloodstream and travel a little bit distance and pop up in different brain regions. So that has sort of evidence suggests that the vascular system may also play a role in the tauopathy. However, in the most of the tau models, that the blood-brain barrier sort of phenotype occur pretty late. So it's really follows after the tauopathy. That suggests that maybe tau, the opposite will driving the secondary changes that potentially be, could be some neuroinflammation or other aspect, and then eventually leads to the blood-brain barrier sort of breakdown. But then it's still open questions. A lot of people are now working on that. So at least for the tau, I would say that we do not have a very clear picture. We know that it may help in the spreading of tau. We don't know whether tau has direct effect on the blood-brain barrier or not. Okay. Again, so I want to thank you all, you guys, for your time and attention. And I want to thank my lab members, particularly the highlighted personnel here, and they basically contributed to all these studies and I talked today. And I don't have time to mention the work from other people's that are driving sort of new development, particularly targeting some new risk genes as well as other models that for us to study Alzheimer's disease. And there's also collaborations that we established through the years. And I particularly want to mention these three persons that I've been working with really closely, especially my mentor, previous mentor, Berislav Zlokovic, and he's still at USC. And our work is really supported, through the generous funding from NIH, as well as, BriFocus and DOD, and then Alzheimer's Association, the Whittier, and then Cure Alzheimer's Fund. So, okay, now I would like to sort of stop here and take questions. 1 second, let me pull out the Q&A. Okay, so we have Edwin asking what happens to capillary when you remove the parasites. Yes, that's a very good question. So let me see if I have the slides. Yes, so we did study the capillary sort of after the parasite ablation. So it turns out that in the capillary, that between those endothelial cells, there's this called tight junctions, junction, cellular junctions. There are tight junctions, right? And there's an adherent junction and other junctions, right? So the tight junction is being destroyed after parasites. And it's also being verified by EM studies. So that really addressed why parasite loss will lead to the endothelium changes and then eventually break down the barrier, because when this is destroyed, everything going to pass through. Okay. And okay. is that relationship between tau and the vascular sort of inflammatory response? I think I don't have an exact answer to that, but I sort of try to address that the breakdown may help the spreading of the tauopathy. And sort of in the Alzheimer cases or in the TBI cases, we also observe is the perivascular sort of accumulation of tau. That is sort of a new discovery sort of in the last couple of years. And that is something that I think eventually should be able to link to the vascular inflammation. So that's something that I think we'll pay close attention to. Yeah. Which non-invasive imaging were used? Okay, now we definitely use the RWD, those laser speckle images, and we use MRI as well, so as a complementary to the laser speckle. And then we also will use the 2-photon imaging. Yeah. Is there a role of mitochondria and let's say the VDAC, which is a calcium channel in the mitochondria, right? And does that play a role in the dementia? Yes. So so that links to a big question: is that the energy deficiency in the Alzheimer patient, right? That can have sort of implication in the neuronal function as well as in the vascular functions as well, right? So the mitochondrial dysfunction is definitely associated with, Alzheimer's disease, even aging, and tons of stuff. And the VDAC is a role there. VDAC drives the mitochondrial dysfunctions that is more defined, better defined in the neuronal dysfunctions, but less defined in the vascular aspect. So right now we do have a project is to look for the sort of, let's say, the mitochondrial defects in the vasculature and in the blood-brain barrier. Okay, now in aging and Alzheimer's, the reason of that is that if you look for the mitochondria sort of level throughout the body, compare all the vascular system of the body, you know that the blood-brain barrier will have much higher mitochondrial level compared to the rest of bodies. And that is probably associated with sort of highly specialized transport systems. It requires a lot of energy, right? So that if there's a defect in the mitochondria, the blood-brain barrier is probably the first one to know. Okay, this is something that we are developing. So hopefully we'll give you guys an answer probably in a year or two. Now, goes back to the VDAC. VDAC is a sort of channel that has been recently found that links the ER to the mitochondria, so that the ER has this calcium store, so that locally when the calcium store releases the calcium, the calcium can go through the VDAC, goes to the mitochondria. That leads to the mitochondria, calcium overload and dysfunctions. And that's something, it's really a new area. It's called the mitochondria sort of, I think, ER contact kind of special contact sites. And this is something that it's been very actively, I think, developing in the last sort of 5-6 years. So, yeah, I don't have the answer for that, but I think that there's definitely should be some new development and towards the VDAC, how that ER dysfunction links to the mitochondrial dysfunction, eventually contributes to the vascular and neuronal dysfunction, and in the age and Alzheimer's as well. So, and so next question is, what are the earliest determinants of the vascular complications, right? And then I'll say, through our studies, we show that there is blood flow changes, as well as in permeability changes. Right now, when we come to the measurement, right, and the measurement usually is limited by sensitivity of the techniques, right? So early on, I think studies that didn't discover that, I would say about 10 years ago, there's not much discovery saying Alzheimer's patient has, vascular problem. I think is that because of limitation of that time is the imaging skills. The MRI usually is only at a 3 Tesla. Now 7 Tesla is available, and that gives us much more higher sensitivity. So And at the same time, the new sort of biomarker, panels have been discovered, right? And those panels, instead of just one or two biomarkers, offers much more sort of reliability, higher reliability as well as sensitivity. So I would think that in the next couple of years, you will see sort of we are going to revisit the kind of this kind of scenarios that which is the parameter best for the vascular complications. And I think in the end is that we have to combine sort of MRI imaging as well as biomarker. So that have become a sort of the standard, at least in the major sort of ADRC centers in the United States. That imaging plus the biomarkers. Right. There's another question about thoughts on the lymphatic systems. Okay, I think I mentioned that earlier that when we talk about the brain circulations, there is a blood flow, but at the same time there's CSF flow that links to the lymphatic systems, right? So let me pull out this slide. So yes, this is a really sort of new sort of area. So that we know that there is blood flow coming in, right? And there's a lot of studies showing that there's a lymphatic system that leads to the ICF, the interstitial fluid going backwards, going out through the perivascular space, right? That's the lymphatic systems. But once these guys going up, then it fused with the CSF flow, and the CSF flow eventually goes to the meningeal lymphatic vessel and fluid drainage, right? This is as I mentioned, is now we should consider every aspect of these into this called brain circulation. Okay, brain circulation should not be just the blood flow. It should be the CSF, SF flow, as well as the lymphatic sort of flow, drainage, right? And this is sort of being summarized through a lot of review recently. Early on, this is a review in 2015. It suggests that the clearance of the the amyloid is facilitated by, let's say, the lymphatic flow, the bulk flow, and throughout also the perivascular drainage, right? As well as eventually now the lymphatic drainage. So all these things. And also occur is the transvascular clearance that we told earlier through the LRP and pickup kind of pathway. So we should all consider all of these together. And then you probably know that Capillay's lab that recently showed that if we block the lymphatic drainage here, there's also increase of amyloid pathology. It suggests that the clearance is should be considered as a whole function of the brain circulations. That blood flow and CSF and lymphatic system all play a role in the clearance. Okay, and I don't have the time, but I do have this one. Is that so? Recently, we started did the clearance assay after TBI. This is done by previous postdoc Yishi Wu. So after TBI, you can see that he injected the tracer, amyloid tracer, fluorescent labeled amyloid, into this region here. Here, I'm sorry, let me. Yeah, so he did a tracer right here and right here. And this side has MTBI, the other side doesn't, right? Now you can monitor how the tracer is cleared in the brain. So in the control side, you will see it travels through the vascular component and goes up to the surface. Now in the TBI, it's now become different. It stays in this area and it's taken up by the neurons. You do not see much of these guys traveling along the vascular segment. Suggests that the blockage of the drainage. This is something that I will mention that this is important for the amyloid beta clearance. Okay. All right. I think we have probably 2 or 3 minutes. So the FPR2, I'm not sure. Can you actually give me the full name of the FPR2? I'm not familiar with this. Anyway, so if you have a question, please send me an email because I'm not familiar with this FPR too. And then, okay. Oh, sorry, I'm not sure. I do not familiar with this one. So I'm sorry. Yeah, I'll try to look up and then maybe we can exchange it by email. Yeah. Okay. Let me check the. Okay, I think these are most of the questions. And again, and really thank IW for organizing this, so we have a chance to share all the studies with you guys. Oh, okay. One more question. So the injection is in the brain. So and these are really sort of the micro injection. We use the sort of the glass tip. The glass tip is very sort of long, and then it's only probably a couple microns, and we can perform the histology afterwards to see whether we damage the tissue or not. Usually, we do not see sort of hemorrhage at all in this injection sites. Okay, and this is because this has been not applied by us, but also by all the neuroscientists for AAV injection. So it's a really sort of micro injection platform. Okay. Okay, all right. Thank you. All right.
Thank you so much, Dr. Zheng Zhang, and thank you all, everyone.